I don't know what the yellowish body is.
It looks kind of like a
Come out for a little bit, video.
bivalve shell with maybe the siphon out, although that would be a strange orientation to have it be sideways and then...
Maybe there was another up from that.
Yeah, I think so.
Okay.
Maxon.
Copy.
Yeah, I don't know what that is to the right of it. Yeah, that does look like it might belong, the white object siphon type of thing might belong to that organism, but who knows what the organism is. Any help from the chat room would be great. Looks like a heterocarpus. Tammy Frank, I don't know if you're referring to what we're looking at.
That was the shrimp from earlier.
The shrimp, okay.
This must be multiple animals covering an underlying rock, like maybe, what do you think, Cheryl?
There was another thing of similar color just above this too. Smaller, but above
Come out
it.
a little video. We'll take a look.
I thought. Yeah, there it is. That almost looks like it
thinking
has
like
a burrow.
Copy. We're not going to have very long to look at this, but
Yeah.
we'll get what we can. Go
Okay.
ahead, video.
Looks like it's burrowing. And it has a siphon too, so interesting. Periscope up.
I have no idea what that is.
Yeah, you
That
can
needs
see
to
that
be
it's been...
excavated.
Okay, I need to push forward. Watch these. Are we good here?
Well, maybe we'll see some more. Keep an eye out for...
Wow, look at that slope. Another couple of squid.
And there's some more of those white... Oh, no, that was eelpout that I'm seeing.
Watch leads nav.
Go ahead, Nav.
The vehicles have gone about 100 meters.
Roger that. Thank you. We've now covered about 100
What's our
meters
final
of our
waypoint? How far away is it?
700 plus track that we're planning to do today.
700 plus.
500 meters away.
Orange thing, crab at the...
Yep, stand by. Okay, go ahead, video.
I don't know if that's alive.
It doesn't look very active.
No. It's like it was a squat lobster of some type before it met its demise.
Could be a molt.
Yeah, it could be a molt.
Our pilot's still looking down about 8-0.
So no one on shore had an idea what that yellowish thing was before? It looked very strange and different.
Yeah, no, we haven't heard from anyone on shore regarding that unusual burrowed organism that had a siphon sticking up. So we're keeping our eyes open for more. This might be one here. No. Anything light colored, I think, might be it.
Yeah, we've been spoiled with perfect visibility for a lot of the dives, and there's so much marine snow in the water column here. And shrimps and squid.
Something white
Eels.
on the lower left. Went off screen.
Wow, look at how steep this is. And darker sediment.
Maybe these are just areas
Do a
where
quick zoom on here.
bubbles
Yeah,
have
sure.
come up
Mm-hmm.
rather than evidence of burrowing. Last night our mapping
Come out a
team
little.
ran one line
Should come
over,
wide,
or
thanks.
several lines over this area, and the sonar can detect differences in densities, and so of course we use it to have that echo of sound bounce off the seafloor because of the greater density than seawater, but it also can pick up in the water column density differences that are made by bubbles, gas bubbles that are seeping out of the seafloor. So we're hoping, and they saw several small plumes across this area. I think those were centralized at the top of this ridge, and they had been identified before by the USGS, but never observed. So we're hoping to find and observe them.
And so the bubble plumes are gases seeping out from the seafloor, and they may have special communities that the gases support called chemosynthetic communities, but not necessarily. They might not have those communities. And so the first trick is finding the bubble plumes, and then the next part is seeing whether there's any chemosynthetic communities.
Coming past the skids video.
And here you see how gravity has its influence. So if bubbles were to reach the surface at the top of that feature, it would cause any kind of disturbance maybe and just detach some of that sediment that would roll down. So we want to keep our eyes open for any of those blob organisms that we saw in the mud. Bob Carney said that it's a mystery to him. He's clueless.
Wow.
The siphon suggests a mollusk, but the egg-like bumps are hard to explain. So collect if another is seen. So we will try to find and collect.
Yes.
Go ahead, video.
Oh, it looks like he's missing one of his
All right.
Yeah.
Gila.
Yep.
It's one of the cancer type crabs.
Missing a critical one. He's missing his front pincher.
And Bob Carney from LSU has been very helpful pointing out some of the critters that we saw yesterday that I wouldn't have even noticed, and
See
many
the
other
barnacles on this guy.
Come on out.
I probably a little off to my starboard.
Copy that.
I can rotate if you need me
wow,
to.
super steep.
See what terrain looks like.
Looks
That's
like
another crab.
Another crab.
We don't need to get it. There's an anemone on the bottom we should get though.
Mm-hmm.
Anemone, just off the screen.
Imagine that has to be a
Yeah, we're
tube
gonna change our
anemone.
movement next time.
Yeah, Nav, it
It's
looks
just
like
completely not matching up.
it's pulling Sirius away from the slope.
Yeah.
Bridge, ROV nav.
Go ahead, RV Nav.
Can you come easy stop, please?
Easy stop by.
Looks like a tube anemone with a lot of his
Go
tube
ahead and
exposed.
video.
Yeah, you don't see that very often.
Sliding down the slope.
Jason Chater from USGS points out that this is
So
more
nice
evidence of
pan up the
small-scale slope sediment transport.
Co-pilot, what direction do
It
you
could
want?
be gravity-driven or it could be
I'm
critter-driven.
gonna
And I was
center
thinking
you real
about
quick.
that when
Yes,
the
please.
crab was walking across
Stand by
this sediment
That'd be
surface.
closer to
Is that a little
west.
fish in
ROV
there
nav
too?
bridge, we're holding station.
Right behind it,
So you're looking
underneath
pretty
the
much-
tube.
Yeah, fish swim underneath the tube.
Just go west.
Trying to get further in.
Yeah.
Can you continue to pan up the tube?
Well, that's one of those little codlings, right?
Yeah, the
Lamanema.
Lamanema.
Yeah,
Yeah.
it's upslope. Just from our perspective right now, looks pretty much west.
So maybe critter initiated
Right.
and then gravity transported.
All right, we'll do 30 meters that way.
Little
Is that a
disturbance.
tongue down at the end there? Come back in video. What's that? Oh no, it's
Oh, that's a
different
nudibranch.
organism.
Or something.
Or what?
Could it be a well-decorated snail?
Bridge, ROV nav.
Go ahead, RV nav.
Can I get a new ship move, please?
Sure.
30 meters,
Or-
bearing
I wonder if it's a little-
0.1.
Is that a
200 meters,
stalk or a
255
foot?
speed
I don't
decimal.
know. I was wondering if it was a holothurian.
I think I see a proboscis on the upper right.
RV
Maybe
nav bridge
two.
move initiated.
Thank you.
Yeah.
I don't see it.
Part of the snail thingy.
Oh,
There it is. Looks like a trunk. Oh. Who's that moving
there's
away
a-
on the right?
the guy making a
It's
run for
another
it.
one.
Okay.
Oh, look at
Oh, it's a train.
Wow.
It's a race.
That's cool.
And look
So
at
it
the
is the muscular foot of a
downslope
gastropod
creep.
that, and
How
then the one on the left
bizarre.
is just covered with sediment and maybe some little forams, agglutinating forams, maybe. I don't know what that is.
Who
It's like
knows?
strings of. But they're marching along.
They really are.
Final
It's
chase
a
scene.
snail race
It
at
is.
the bottom of the ocean.
Final chasing speed. Oh, that one's
And
drifting.
just look at that creeping sediment down slope. It's
Yeah, it
going as
does.
slowly as the snail.
Does kind of start pushing. And
great.
Jason Chater said that the bioturbation triggered by sediment transport is something we haven't constrained well
Okay.
yet. And Jill Bork was the first one that noticed that that was a snail.
Looks
That's
like there's
amazing.
some siphons sticking out of, well, not siphons, maybe just tubes sticking out of the sediments above.
Oh, yeah.
There's several of them.
Video's clear.
Great, come on out.
Those are funny.
Could we pan a little bit left when we come out to see if there's a white bacterial mat?
Panning left. Where, based on lasers?
I don't see it. I think that we would see it by now if it were here. Okay, so
This is going to be better. I'll be able to go up slope.
It just went off screen left.
black reduced mud is what we probably are going to see over here.
Yeah,
Yeah.
I think what they were thinking is the mat is going to come in your screen on the left.
Still further left?
Yeah. It's kind of right on the edge as he came about.
I don't know if these white particles are organisms. Maybe we could take a quick zoom onto the.
Yeah, this would be
They
a
may
good zoom
be-
here.
Go ahead, video.
Maybe precipitation. Or they may be
I
shells.
think they're just shells.
Yeah, they are shells.
Is
You
that
want
some of that
further look,
gastropod?
or should we come out?
No, that's fine.
Okay,
Thank
come
you.
out video. I am still not seeing this mat yet.
Yeah, that's
Further
okay.
over.
Maybe just
We can continue on up.
turning your head. I don't know. It was small. It had been just shadows on that.
I think so.
It's steeper here.
Rolling.
Ooh, this is interesting. Might be where those little mud balls came
Ooh, and
from,
there's
something like that.
There's a squat lobster.
Rolling, that lid opens up. No, it's not.
It's gonna
push
pinch it.
Closer. It's gonna pinch it.
It's gonna.
Wow, there's some reduced sediment.
Where are we in that ship move right now?
About halfway.
Copy.
Something was moving. It's just directly
This guy right above us?
Yeah.
Yeah, I saw that. Let me get a good view on him.
Oh, it's just a couple of pout eels.
All right, laser's coming out.
Or maybe just one with a...
Curve curling all up.
Oh, gosh, I thought that was something growing on him. That's just his fin. We haven't seen it expanded like that.
So Martha Nazinski was asking if anybody knows what species that snail is, because she hasn't.
We got two things up here.
She's seen them with hermit crabs inside, but never with the live gastropod. I wonder if that would be something worthy of collection, but if they're going to be really small.
Yeah, we were lucky
Maybe
to
scoop
see
it
it.
if we could.
Give it a
wow,
try.
this is
I
a
don't think
beautiful
it's gonna
squat
do
lobster.
anything.
I know.
Laser
Go ahead, video.
coming out.
Well, it appears as though he made a burrow. Do they burrow?
I haven't seen them actually digging a burrow themselves, but I've seen plenty in burrows, so I imagine.
You want to?
And I think
fire
there's
up my,
a
swing arms?
black belly
my
rosefish
hydraulics right here.
too.
Yeah, hydraulics are, you're up.
Okay.
Pilot to the left was a fish. He may not be there still, but
Copy that. We're
All
gonna
right.
try to move this eel.
oh.
Got sick of looking at it.
I'm up and ready to go.
Copy. Do you want camera view?
I'm just gonna use these manips. Yeah, if you wanna just follow me over with the mini.
Mini? You got
Yep.
it. Heat. 2. Alright.
Okay, coming on.
And this squat lobster species is Munida valida.
Alright.
And right now we're trying to free the snipe eel from the rock box.
You can look at camera 3 to get our multi-view.
It's gonna be the best way for this.
Close your jaws and open it like normal.
Oh, you just want me to open the box?
Isn't
Oh,
that
okay.
what you were trying to do?
I was gonna try to like brush him away,
Oh.
but yeah, I can do that.
If he didn't come off when we did our
So
turns, I
Jill
don't think he's just going to brush
Bjork
off.
from
I was gonna pull
USGS
it
said
off.
that snail might be family Bucinidae, possibly genus Colus, and that they've picked them up in trawls from the
here we go.
Mid-Atlantic
We'll
canyons.
just open it and see what happens.
you're pushing in on the box.
Yeah.
Look at your mini suits, that'll give you the best angle.
Yeah. I'm hitting.
Hitting a limit somewhere?
Yep.
Yay! He's free. Good job.
That's really great.
Nice.
I wonder if he's still
Follow
alive.
you, pilot.
Looks
Okay.
like he might be.
Yeah, looks like he swam away.
Nice job.
The docks are secure.
Can we recenter HD2?
HD.
ROV
Oh, sorry.
HD2, sorry.
Yeah.
Okay, gonna push out. And you're caught up now. Perfect.
Oh, some
Look at all
nice
these guys.
squids. Yeah.
Actually, let's
They
take a
wanted
look at
that
this.
fish.
Yeah, yeah, I forgot. Oh boy, that's a that's a bigger one, huh?
Big squid. Yeah. He's
It's
inking
just-
all
Wow.
over
All
us.
right, get out of here. Okay.
He's still there.
Goodness. Yep. That's about to happen.
And I don't think I've ever seen a black-belly rosefish in sediment like this. This is kind of unusual.
Lasers
Come on in, video.
Lasers
Could be
off.
lost, or maybe.
We saw so many of
This'll
these
clear in a
yesterday
second.
in a little rocky landscape at similar depths. A little shallower than this, but a little out of place. We saw one at the beginning of the dive, too, on the flatter portion.
Yeah, that's right. When we first sat down.
Little mini
Full
avalanches.
specimen.
They're great. You think of each grain just dropping a centimeter or so, but you amplify that by hundreds and then thousands of years, and you can imagine how erosion affects this larger area significantly.
Video's clear.
Okay, coming up.
And how these canyons are formed.
There's something else over to the left here. Let me go take a look.
Lasers coming on. Lasers.
Ooh,
That's
this might
what
be
we were looking for.
That is
bacterial mat.
definitely what we're looking for.
Okay. Just give me a moment here. Actually, what is this right below us too?
Is that not your skid?
Quick zoom in there.
Oh, and there's another one of those. Oh, wow. I guess that's another snail.
It's a
And the
crab.
There's a
holes. What are
crab?
these holes
Yeah, that's
too?
a tiny crab.
Oh, yeah.
It looks like a
One of the
young cancer.
cancer genus. Tiny, though.
It's like he just came out of the hole covered in sediment.
Okay, going up now to look at the main event up here.
Another snipe eel. Yeah, so
Come on in.
this white stuff is a bacterium that is often associated with methane leaking from the seafloor.
A little tilt up. Show
So
some
we
more.
should keep our eyes open for a bubble plume.
Continue to the top.
Not seeing any bubbles around the edges. Here, watch leads.
Yeah, I wonder if
Go full zoom. Full
Go,
specimen.
go for it. I see the little holes there on the edges, though. Do you see on that bottom right of it?
mm-hmm.
Seems like the top one is the brightest. Go to the very top edge.
Top left.
Yep. Max zoom.
Co-pilot, you ready for another move?
Yeah, I think we can do one. I'm looking at pilot at 6-0.
Video's clear.
Come on out.
Still like this direction? 260?
So pilot is looking at
I'm looking at 300, and I'm looking up slope.
Directly up slope.
So,
Lobster upper right.
Cheat.
I think if you look at high pack, I think this ridge
Yeah, I
that
think
we're
that squat lobster
we're
is
supposed to
actually
be
the
going
one
along
we've
might
been
be
seeing.
offset
He's moved
a little.
up slope with
I
us.
think so too.
So yeah, we can head up this ridge if you guys want.
That was
Get
a
to the
Munida
top.
Valida.
That makes
Go
flying a
go
lot
high
easier.
and then turn left.
Perfect.
Yeah, as long as it
Okay.
still is good for the goal of the dive.
So-
that's fine. Let's just rotate a little starboard then at 300.
Yeah, go for it.
Nav, you were saying?
Go ahead, watch lead.
More bacteria.
Yeah, I'll go take a look at it.
Roger that. Will do.
Get it between your skids.
I'll do what I can. Gotta make sure it's safe first.
Understood.
True that.
Nice cover up.
Quick zoom in here. Just to see these holes. I want to make sure nothing's coming out of them.
So
Looks
bivalve.
clear.
Is
or
that a,
something
yeah,
to the
bivalve
left.
to the left?
Mm-hmm.
I was actually going to get a little bit closer here. Watch these. That was just to check to make sure
Okay.
it was a good spot.
If it looks like necrotic tissue, it's got to be good.
Well, maybe that's why we're seeing so many pout eels, as they like this particular environment.
Okay, now I can come in video.
Didn't see that many in high densities before.
Do you want to see the bivalve as well?
Sure.
Okay.
Almost looks like a brachiopod.
Mm-hmm.
It's kind of a weird orientation for a
Ready for
bivalve
that
that's
new
still
co-pilot?
alive. It's like
Yeah,
attachment up.
Okay, 30 meters, 290.
copy that.
Wow.
Bridge, I'll be nav. Bridge over.
So Cheryl,
Yes, so ship
maybe
move,
this is a
please.
good time for you to
Go ahead.
mention
30
about
meters,
these bacterial
bearing
mats and
290
how
degrees at
they
0.1.
form from the gases.
Yeah, so they are actually
Understand 30
able
meters
to kind
bearing
of
290
change the
speed decimal 1.
Good copy,
methane
thank you,
and
Bridge.
gases that seep out of the seafloor into a food that others can eat, other organisms.
And it
Video's
serves
clear.
as the
Come on
base
out.
of the food chain or the food web for this environment for many
Don't
organisms.
see any shimmering or anything.
None at all.
Copy. All right. I'll push out a little bit. Oh, look at this guy. Well, we've seen them.
Watch leads, this is nav.
Go ahead, Nev.
ROV nav bridge, move initiated.
Thank you, Bridge. I had a question about where the bacteria for these seeps originates. Meaning, like, is it coming up with the seep?
No.
Or is this bacteria that just happens to find a small seep like that?
Yeah, so it definitely wouldn't be coming from underneath the sediment. It would be something that finds this habitat and then multiplies. And bacteria can multiply very quickly.
Oh, there's a eelpout sliding down
Oh,
the
no.
slope. Oh my gosh.
It's going sleigh riding.
What?
Well, this is another.
What's the name of that fish again? Not the
Go ahead,
eelpout, but the
I'm gonna
this
kind of drift
thing
by
I
him real
keep
quick.
This is a
lip-
Lamanema.
Lamanema. I
Wanna
cannot
Stop, do we?
get that in my head.
Well, I'll give you.
And I think that this one is Barbatulum.
And Nav, we didn't mean to laugh at your question, but it is sort of the $1 million question.
Yeah,
of,
exactly.
Of origin of life.
The chicken or the egg
The
kind
chicken
of
or the
question.
egg, yes. That
Sure.
for so long, photosynthesis was the only way that scientists knew of life being originated, of primary productivity at the surface, which requires light, of course. And ever since discovery of hydrothermal vents back in the 70s, started realizing that chemosynthetic life could originate on the seafloor using these chemical reactions with gases and the production of bacteria. So,
Well, thank you for the answer. I'm just surprised that the bacteria is able to locate these seeps in a sense.
Yeah.
And I imagine it's kind of just blowing through the water, but
What was that, Kova?
And that's true
I
for
have a
all of
direction
these
change.
chemosynthetic
Yep, looks like we're getting to the
communities, is
top
how
now.
do they find a
great.
new site?
we're learning with exploration of a tremendous biosphere of
All right, looks like
bacteria
we
just
found the
below
top
the
of it,
surface,
maybe.
not just here, but globally.
Look, it needs
Associated with
cleaner water already.
seafloor
Oh,
spreading
looks much
ridges
nicer.
and the heat that arises from those, and
Wait, let me
hydrothermal
just take
vent
a look in here real
systems.
quick. Quick zoom video. Something's. It's just sediment.
Yeah,
Now it's me.
that's more sediment.
Yeah, there was just sediment there.
Oh, wow, I didn't realize this. Bob Carney said that microbial chemosynthesis was recognized in the 1800s in shallow systems.
No kidding.
I didn't know that.
I didn't either.
That's a big hole.
It's an accurate statement.
There's the hole at the bottom of the sea.
Someone's digging their way to China.
Wow.
Oh,
There's
there's some
look at
bubbles.
that.
Yeah.
Slow
Nice
trickle, but
job.
there it is. Wow.
That is not a fish burping.
There might just be some like gas trapped underneath here, and I jostled it.
I don't
Huh.
think so, not with that much sediment going. Can we just see what it's going to look like with the extra
Oh,
lights?
that was
Go
fortuitous.
for it.
Wow.
All right, fog lights are coming up.
Our first
EQ1.
seep.
It's too red.
Kill lasers
That's right.
We can lose the extra lights.
coming out.
Cheryl or Leslie, were you there earlier today when Kevin was talking about they'd
Sit
mapped
here and
bubble
get a few
plumes?
bubbles if we
Yeah.
And
tilt
just
to
as
the
they were
lower
turning
lip.
off the multi-beam and turning and
The
like getting in with the
lower lip?
Yeah,
ROV,
tilt up.
like
Tilt
the
up.
it turned off, so the ROV couldn't find the bubbles. And then later in the
So
day
that
when
the
they were done with the
bottom- yeah,
ROV
yeah.
dive and they
You just want to see it.
got back
go
into
in the water
multi-beaming,
column.
turned back on again. So like within like hours, it's like
I feel like I
very
jostled
frustrating.
some.
Yeah, so I wonder if we'll see more of this as we continue on. It's a very
You wanna give
slow
us more
trickle of
thrust then,
bubbles
since
here.
the Jostle it again.
That's exactly what happened. There it is. Yeah, it's directly
There's
under me.
another
I'm gonna
Oh, it's
actually
not
move
coming
back
out of
over
that.
there.
Okay.
Might be a whole lot under me.
Yeah, I wouldn't be surprised if there's more than one.
Mm-hmm.
Oops, we might have unearthed
Hey, there you go.
some
There's
of
some
them.
more.
Oh dear.
Well, what I'm wondering is you're releasing it, because as you push forward there,
That's
you kind of
what I
compress
think.
the sediment.
Yeah, yeah.
That's what I
Same
think.
with your toe as you
Actually,
came out.
take a look at this sediment too.
Wow, yeah.
Like, you want to zoom in?
Very dark, reduced sediments.
I wonder if this sediment is kind of saturated with methane.
Come on out, video. Let's.
Out.
Yeah, I just want to see if there's more there.
Hi, Cheryl. This is Carolyn.
There's a little bit.
joining.
Right
Hi,
down here,
Carolyn,
too.
how are you?
Hi.
We have Carolyn Ruppel from the U.S. Geological Survey in Woods Hole online, and she has spent a lot of her career working on locations like this.
So what are your impressions, Carolyn?
Well, I've been watching sort of for an hour now vicariously, while I was on this other project review.
This
and
whole field's got some
yeah,
of
I
it.
guess I'm hoping to get to the actual
see that
more
video? There's a whole lot of
feature
them.
which I think might be upslope. I think you guys also think that. That one we just saw, though, I'm actually just wondering whether someone mentioned it looked like an excavation that had maybe disturbed some gas that's very shallow beneath the surface. And Jason Chater and I were texting back and forth, and obviously there's a lot of reduced sediment here, so that would imply you probably do
That's
have
just
a lot
it.
of
You're already covered with mud.
oxidation
Okay, here we go.
going on right below the surface. So there's probably a lot of methane here. You just have to access it through these seeps
It's
or
pouring out of that
excavating
one.
a little.
So,
If you tilt down, you see it came out from under you.
Yeah.
yeah, it's coming out of the little holes when
yeah.
when
Yeah,
the
we're
sediment around
definitely
it is kind of
going
Check these
to
little ones up there.
try to get to the top where the probably larger plume was that you
Yeah, I
gave
think
us
it's
the
time to-
location for.
I can push. I can
And
go.
but it's exciting to
You're
see this
changing
down
directions.
here also.
Oh, yeah, definitely.
bridge, Harvey Nav.
I actually, Cheryl, meant to send you an
Can
email
we,
an
before
hour
we
ago.
take off,
One thing that I found really
Go ahead,
interesting
Sort
as
of
a
get an overall
non-biologist
complete.
and having
we
watched so
actually
many
have
of
5
these
more
dives,
meters
we
left.
did come
Let's
across
just
a
end
place
it right
where
here
there
if you
were
would.
What was that
all these
video?
sort of
Once you
shallow
lose the
areas in the
background.
seafloor, were each occupied by an organism. And those of us who've watched a lot of these dives, these little shallow
sure.
trenches that you guys always tell us are made by the various biota, I thought that was really
Partial
telling.
turn.
Because I had
Thanks,
really
Bridge.
seen like the organisms actually occupying these little dugouts. And so I thought that was terrific.
Yeah, that's very true.
Ship is now easy
You can
stop.
see now as we're turning around, you can see lots of little holes in this particular section.
Yeah, there are lots of holes, and I thought that they were burrows, but who knows what if some biota caused them, or if the bubbles cause them, and then the biota uses those little crevices.
All right, let's find your alignment with the ridge so we can keep.
All right.
Pushing.
Looks like we got holes all up and along this.
So Bob Carney pointed out that the bacterial mats are typically sulfide dependent. I guess we just don't know whether it's methane or whether there's sulfide also, or.
Take a look at this
Well,
guy,
the
too.
anaerobic oxidation
Actually,
of methane
Quick
has as
zoom
one of its
here,
byproducts
video.
of sulfide. So, it's all connected
Nope.
system,
Wasn't what I thought.
right? I think I said that
Bubbles.
right. Bob Carney will say in the chat room if I said it wrong.
You want to see the crab or the starfish?
The sea
I thought
star.
the white thing was maybe one of those white things from earlier. Actually, I think that's a shrimp, not a crab.
Some trace marks from some other organism, it looks like.
Mm-hmm.
Come on in video.
So just to clarify what we just said, so Bob says I'm right. So there's usually a sulfate methane transition in the sediment. So methane is excluded in the very near surface sediment because it's already been oxidized. And one of the oxidation products, anaerobic oxidation of methane, oxidation without oxygen, is that you're going to have sulfide there. And so that's why the sulfide and the methane kind of interplay at these places. Sometimes the methane comes all the way to the surface, and you have no sulfate reduction zone beneath the surface. But we're here, obviously, we have a lot going on. We see oxidized sediment, the darker sediment strip beneath the surface, and then potentially sulfide-dependent organisms, perhaps including the bacterial mat.
Well, that was a great explanation. Thanks, Carolyn.
That's better. All right.
Go over there on the right, bottom right.
What do you guys see?
Casey Cantwell has asked me in the chat room, this is Carolyn again, I'm sorry, to say a little bit about how we know about what's going on the East Coast with these methane seeps. So prior to about 2011, we only knew about 3 locations with methane being emitted from the seafloor along the entire U.S. margin. And some of those were over features like salt biopiers. Those of you who have watched a lot of dives have seen, you know that we talk about those in the Gulf of Mexico. They're far less widespread on the Atlantic margin and in the southeast part down the Blake Bridge, where there have been some dives on this particular expedition as well. And so starting in about 2011, NOAA Ocean Exploration started finding, and it wasn't because they had just turned on, it was because they always existed, but we had the tools to find, started finding water column methane plumes. And NOAA and the USGS talked quite a bit about that.
I just want to reference myself
2013. In 2014, a paper led by Adam Starkey with myself and some other NOAA and USGS co-authors, including Mimi Lobecker, who's one of the leads for mapping on a lot of these cruises, published a paper in Nature Geoscience that talks about all the methane plumes that had at that point been found on the U.S. Margin. And so the plumes are what we can image. The plumes, in terms of diving at the base of them and finding the actual seep on the seafloor, we sort of had a 100% success rate with the few dives that occurred until then. In the end, what we published was about 570 methane plumes that we traced into the seafloor along particularly the northern part of the margin from about where we are today, north to George's Bank, which is all the way out.
It's getting a bit easier up here, actually.
And some of those were diffuse, and some of those were discrete. Some of them were very obviously plumes that could be traced into seeps. Others, it was harder to say. Since then, I've been involved in 5 or 6 cruises on this margin. And one thing that the USGS did a few weeks ago was provide NOAA with a provisional updated database in this area with new plumes that we had identified in the interim based on various cruises that we had had. So I think the point is that anytime you go out with these you find more plumes. Sometimes we find places where we had identified a seep and it's no longer leaking. Other times we find new ones. But we definitely, this is a very leaky margin right now, as far as we know. And this has totally changed our perspective on what is supposed to be a cold margin that shouldn't be very exciting. In fact, it's leaking lots of methane almost everywhere that we go in certain types of locations. Since we're still over seafloor, this fairly featureless, let me add that in the original paper in Nature Geoscience, we linked the position of a lot of these seeps, or plumes, which is what we imaged, to the up-dip limit, the landward limit of the stability field for methane gas hydrate, which is an icy form of methane in water that exists below certain pressures, at greater pressures and moderate temperatures. And that on the U.S. margin is about 550, 575 meters. This exists in the sediment. It's naturally occurring. And when it's perturbed, the temperature heats up, it can break down.
Large hole
and a lot of
right.
the seeps were just up-dip of that location. The work we've done the last few years, we're not really sure that the seeps are connected to the degradation of this ice-like deposit due to what's known with the warming waters on this margin. But there's still a lot of methane coming out.
There's something up there, too. Do you know what that is yet?
We're still working on sourcing
Looks
that
like another
methane.
hole,
Does it
just
come
darker.
from methane hydrates? Does it come from
Separate
other
tunnel.
sources? And does it come from
Let's take a look.
microbial gas that's produced in the sediments, or does it come from processes like those that occur in the deep Gulf of Mexico when you heat up organic matter, thermogenic methane? And I'm going to stop there because I'm sure the leads on the science on the ship want to now say something more interesting.
That was very interesting. Thanks, Carolyn, because we will certainly have a lot of questions about this area. So I hope you'll stay close to the phone. I'm sure you have other things going on, but your expertise is invaluable, and we really welcome for you to call back at any time and stay on the line with us.
Science, that darker section, that's most likely not rock, right? Could it be some different shading of biomaterial?
That's the oxidized sediment that we're seeing, so it turns dark. The reduction turns it dark, basically.
Thank you.
Amanda Demopoulos says, that's my kind of mud.
Look at that hole at the bottom right. The light's going in.
So I wanted to relay several chats that went on recently that I think will be useful for many folks listening to hear. Zach Prue, a graduate student at the College of Charleston, asked of the scientific group here, do the bacterial mats we saw earlier grow directly on top of methane seeps or just in the vicinity? And Tara Luke commented, as long as there is enough methane around, it could be either. And then Bob Carney said, bacterial mats typically are sulfide dependent.
Down there.
And it just jumped off my screen. Sorry.
That's just sediment getting reflected by the light.
He also said, seep is a useful, ambiguous term. Bubbles, brine flow, or just black sediment. We are probably seeing active and inactive bubble-made holes. So downslope, we were looking at a lot of these, what we thought might be burrows, and started seeing that underneath this oxidized brown layer, we were seeing that darker, reducing environment evidence in the black sediments. So maybe almost all the, quote, burrows, unquote, are formed from bubbles at some point in their history, and some of these have filled in. You can see depressions now in the screen that maybe those were former seep areas where bubbles came out to the surface.
You had to? Oh, okay. What's going on with this guy?
And we're seeing how the telepresence capabilities that we have are very, very helpful here. We're able to have people on shore see this and communicate to us via the chat room. Is that an anemone?
I was wondering if it's a tube anemone.
Looks like a tube anemone, possibly.
It's got kind of striped tentacles, which we haven't seen before.
We can get a bit closer look there then.
And we've stopped seeing the eelpouts that were so abundant downslope.
Mm-hmm. We're now at 441 meters. Another black belly rosefish.
Go ahead.
So we've climbed about 50 meters so far on this dive.
I think we're good.
It does look like it has the really tapered tentacles of a cereanthid and the different tentacles in the center.
Go ahead, Nev.
We've gone another 100 meters.
So we've gone 200 meters since we hit the seafloor today. There's another Laymanema on the left side.
And we've climbed over 50 meters. So that gives you a, you can picture that in your head. You can see how steep it is.
Yeah, it's very steep.
There's a little trail here.
Interesting track.
Looks like
I
a
think that's made
crab.
from one of those crabs from before.
At least ghost crabs leave
Something on
that
the
type
bottom
of
right. That was grabbing my
trail
attention.
in the sediments.
Nice jelly in Sirius's view. Big one.
Jellyfish is almost as large as D2.
That was very fortuitous that we saw a few bubbles come out of one of those holes, so we know for sure that that's what's causing some of this pitting and the reducing environment as well. Yeah, I think that's a sideways scampering crab that makes that tracks.
That's like a shallow water jelly.
Oh, in the Sirius?
Yeah, I think it's like a chrysaurid.
Yeah, we're looking at camera 2.
It's not, definitely not one I typically see in the water column.
By
Like
shallow.
down this deep.
Yeah, by shallow, what do you mean?
Like from the surface down to maybe 50 or 100 meters.
Okay.
But yeah, I usually see them like just standing on the bow from the, just looking at the surface. I don't usually see them down here at all.
It's another anemone tube. This is a good angle.
Here's
I guess it could be, I
another-
mean,
the
yeah, it could
anemones
be
maybe.
carried down here by currents, or maybe I just haven't observed them deep, but that is a normal thing. I don't know.
Go ahead, video.
Yeah,
Another,
you were right. Tube.
It's a beautiful shot of
Yeah.
Serianthid tube anemone.
And you pointed this out earlier, Cheryl, but in previous dives when we've seen these Serianthid tube anemones, we've seen the set of dark, deep red ones. I haven't seen these ones that have like sort of striped tentacles.
Yeah, Scott said
as
that
much.
there were some yesterday that had striped tentacles, but they were a different color too. So
That could be just depth differences, you know, species distribution by different depths that we've been diving on.
Video's clear.
Come on out.
Another tube up ahead.
wow, it's still really steep.
I think he's got his mouth wide open.
Go ahead, we can do a quick zoom. That might be different, isn't it?
Oh wow.
Oh yeah.
A very different looking tube anemone. And a little, I don't know if that's a Bathinectes crab underneath the Serianthid. That's a swimming crab, a portunid.
Maybe that's the tiny one that we got yesterday.
Oh, you got something.
Saw
Look
some.
at that.
Oh.
Caught him.
Wow. So here we have a predation event.
Lots of thrusters.
Looks like an amphipod
Yeah.
is getting eaten by the tube anemone. I've definitely never seen this happen. Wonder if he'll get away.
He's not done yet.
Yeah, it looks like a Hyperion amphipod. Obviously, it's mostly translucent, which to me means it's, yeah, a typical water column, typically traveling around on a jelly.
And then it gets caught in these tentacles that have stinging cells.
Thank you, Bridge.
And so he's probably going to wind up being lunch for this tube anemone.
Nice timing for zooming in on this particular anemone that we got to see this.
He's trying really hard to get away.
Oh no.
And so what this anemone wants to do is get him. Oh, he might be free.
Man.
He's trying to get him into his mouth there in the center, in the middle of all those tentacles, the ring of tentacles. But he's still kind of stuck. Yeah, so the nematocysts are the stinging cells.
Oh, he got away.
And the spirocysts are the sticky.
Oh, he might get taken
away.
by the crab.
Out of the fryer and into the
Oh, the
frying
crab's
pan.
afraid of him.
I think the stinging cells in that anemone has affected him.
Yeah, I wonder if the stinging cells have done a number on him and he's not functioning as usual.
I was actually just thinking that maybe the
That crab
stinging
has
cells
him
of the
now.
anemone
Lift
couldn't
up
penetrate
and
the exoskeleton
Lift up,
of the
I'm gonna go
amphipod,
flying
and that's
away.
how he was able to get away. But then, you're right, he didn't seem to be behaving normally after his escape, so he was
Crab is pushing
partly
him away.
affected.
This is probably a gammarid amphipod.
Crab's, from this point of view, it looks like the crab wants nothing to do with him.
Mike Vecchione pointed out this is a good example of benthopelagic coupling at baffle depths. So you have this amphipod, which is usually in the water column, but during the daytime
All right.
they will
That's the one
move
that got away.
deeper
Oh, he's
so
coming
that they
back.
can avoid predation. Of course,
He's-
there are other predators deeper.
Oh no.
Yeah, what Scott's saying is that
Okay.
it looks like a gammarid amphipod instead of a Hyperion amphipod, which
Move on.
generally means it would not be one living in the water column.
Oh, okay. It still is really not doing well.
Yeah. To me, it looks kind of like Themisto, the
Uh-oh.
genus Themisto.
He might be.
That is actually a water column one. So that's interesting. I hope.
And we were also
Okay,
talking in
yeah.
the chat room here about the pit that is formed around this tube anemone and whether the presence of the tube makes these sediments scour a bit.
Or if it's this crab
Oh,
that's clearly.
The crab almost got
Living-
the amphipod. Yeah, that would be a good position. Oh, he does have it, doesn't he? That's a very useful position for the crab to stay there in that pit and just take the leftovers.
Yeah. So yeah, it's a good question as to whether it's just the water current scouring around the tube or whether this crab is making an excavation around the tube.
Crab doesn't want him in there. All right, video, you clear?
Very much so.
All right, let's move on.
That was a pretty awesome multi-species interaction there.
Yeah, so the tube anemone
Is
had
he?
an
Let's go back in.
amphipod.
It's a
It
different
was stinging
subject.
it, holding it and stinging it, and then the amphipod got away, and it was obviously
Yeah, but now
affected
it's
still
someone
and
new.
not
Someone new is now
behaving
in there.
well. Oh yeah, there's Oh, he got it again.
No,
Is it-
different one.
Oh,
it's
he got a different one?
Okay, so
I don't see the
then
one
the
down on the
crabs
crab
Okay.
anymore.
It's the same one.
It's
amphipod
there.
that the tube anemones didn't get, and now we have another